So what I'm going to try to cover is something, first of all, about clinical trials, and whilst we all realise how important they are, just to remind ourselves why they are the cornerstone for advances in evidence-based medicine. I'm going to give you a few examples of how clinical trials can be practice-changing and to lead to advances in healthcare, and then look at the changing environment and how we need to tackle that in terms of where we go next in terms of clinical trials, both in terms of the rising costs of healthcare and trials, new technologies, changes in trial conduct, and challenges that we face if we're trying to personalise or individualise treatments to individual patients. So some examples of clinical trials which have changed practice that I've been involved in. One goes back a long time now, is the use of statins in randomised trials to save lives and prevent cardiovascular disease events. And this is now used routinely worldwide for many millions of patients. We were involved in led the lipid trial using pravastatin, which showed survival benefits for patients with average cholesterol levels. And that's now been recently followed up with more than 16 years of follow-up, showing that those initial benefits are maintained long-term, so that with a very excellent safety profile as well, in terms of no increase in non-cardiovascular deaths or cancers. Aspirin is a treatment which is used for a lot of things, but it's also now been shown to be a benefit in reducing recurrent venous thromboembolic disease. And whilst the standard therapy for that are oral anticoagulants in terms of preventing recurrent events, for those that don't take them or they're no longer suitable, aspirin provides a very cost-saving and effective treatment for those patients, both here in Australia and worldwide. Clinical trials are not just about drugs, they can be about surgery, healthcare delivery, a whole range of things. And to illustrate one of those, the use of giving oxygen therapy to preterm babies in terms of whether to target higher or lower oxygen saturations. A trial led by William Tano-Mordi, the BOOST-2 study, and which recently got the runner-up for the ACTRA Trial of the Year award, is an example of something which has had a substantial impact on affecting lives of babies in the future. So that particular one, the BOOST-2 trial, looked at higher versus lower targets for oxygen saturation, where prior to this and a number of other trials done internationally, it wasn't clear whether to give oxygen within the range of 85 to 95% saturation, whether to aim for the higher or lower end of that range. So that range was regarded as within the options of standard of care. So this trial and 4 others internationally looked at giving the oximeters offset to give a slightly higher or lower reading, so in order to be able to therefore compare in a masked way whether the higher or lower targets achieve better outcomes. 2 of those trials, the BOOST-2 trial and the one in the UK, were published last year in the New England, showing significant benefits in terms of less deaths occurring for those getting the higher oxygen targets. And that has also now been looked at in all of the 5 major trials, these 2 and 3 others, showing that those benefits in terms of lives saved for the higher oxygen targets is clear across the 5 studies. And that's going to lead to addressing a question which was unanswered for over 50 years and is going to have a major impact on the future preterm babies around the world. So that's just a few examples to remind us that clinical trials can make a difference, and it's the cornerstone of why we do the trials, to improve healthcare and to improve clinical health outcomes. We're facing, though, a number of challenges, and the first of those is that there's increasing costs of healthcare and of clinical trials. So one question people often ask is, well, trials might be a good idea, but if they're so expensive, can we afford to do them? The other way of looking at it is, can we afford not to do them? This is the estimated increase in government healthcare expenditure in Australia, first from the 2010 intergenerational report and then more recently updated, where it's projected that over the next 40 years, healthcare costs are going to double, and that that's going to see by 2055 more than 5% of the GDP and more than $260 billion spent on healthcare expenditure just from the Australian government. So if we're going to be spending all this money, if we can use it wisely and spend it on the things most likely to benefit, then that's a great idea. And clinical trials is one of the ways we should consider of being able to do that. So are clinical trials, in fact, a good return on investment compared with spending money on healthcare? A recent report, which has been presented but not yet published, commissioned by the Australian Clinical Trials Alliance, looked at 25 clinical trials undertaken across 3 networks across Australia, and demonstrated that for every $1 invested, there was an estimated return of $5.80, so that spending money on clinical trials leads to money saved, either in terms of other healthcare costs avoided or in terms of health benefits in terms of additional quality adjusted life years, where this model assumed $180,000 in terms of the value of each life year gained. Now, even if you assumed it was less than that, it would still be a great return on investment. In terms of other trials, the lipid trial, which I mentioned earlier, showed that pravastatin reduced mortality for those with coronary heart disease. And this particular trial led to changes in the pharmaceutical benefits. And the trial was estimated for the use of the treatment to cost $6,300 for each life year gained by using this treatment compared with not. Now, the actual trial cost about $40 million. And it impacted on millions, and other trials has impacted on millions of patients worldwide. So if we look at the costs of the trial and distribute that across the people who have been treated, that $6,300 would still be less than $7,000 per quality adjusted life year for the trial and the treatments introduced. So this trial has been far more cost effective than a lot of treatments that we use today. NIH had looked at a program of studies in the US which looked at treatments for neurological disorders and stroke, and showed that across 28 trials costing some $335 million, that the benefit of this would be roughly $7,700 per quality adjusted life year. So these are examples of where clinical trials have been good value for money. The last example I give of that is the use of aspirin, which shows that in the study I mentioned earlier, there is a benefit of about a 40% reduction in recurrent events when you take into account for those on treatment. And if you look at on the intention to treat analysis, a 1/3 reduction. So if you treat 1,000 patients each year, you might prevent 20 to 30 recurrent events at the cost of 3 extra bleeding episodes. There's also a cost saving, though, for this particular treatment in terms of the cost of treating those recurrent events. And so with every 1,000 patients treated, it's estimated you'd save about $200,000. So this trial, which cost about $4.5 million funded from NHMRC, would more than pay for itself within a year or two of the treatments now being used for patients in terms of having aspirin for this particular condition. What does this mean, though, for future trials? Well, what it means is that we should be thinking about not only do we fund studies which are high-quality science and which are innovative studies, but those that are likely to impact on future practice and policy, and those particularly that are a good return on investment. The examples I've given you are after the fact. We now know that they were good value for money. How do we decide on the next study we're going to do whether it's going to be good value to money to invest in it? And the use of value information analyses is a way to try to look at that problem. And I believe that this hopefully will become one of the cornerstones for decisions about funding major trials through MRFF, etc., is using this kind of methodology to ensure that our future clinical trial research is a good return on investment. The costs of treatments are going up, and this is particularly so for cancer, where the costs of cancer drugs, some of them now in the US, are over $60,000 per month of therapy, which is a treatment that very few people would be able to afford. So if we're getting into a situation that treatments are becoming unaffordable, then what are we going to do about it? Well, why are these treatments becoming so expensive? One of the reasons is because of the cost of the clinical trials we need to do to evaluate those treatments. If we look at a lot of treatments with 10,000 compounds, one of those might lead to an FDA-approved indication, and you need to look at the costs of those overall programs of the early basic science work through to the clinical trials at the end. If we can be smarter about this in the future, then hopefully we can reduce some of those costs. But at the moment, they've been doing the opposite of going up. So some of the ways of trying to do that is to consider targeted therapies as a way of giving treatments which are more directed for particular patients. And in cancer in particular, but also in other areas, can we look at the molecular profile of patients based on genomic or other information, which might be a better indication of which patients might benefit and those that don't. So some of the ways that you can undertake those sorts of studies is to consider enrichment designs where you recruit patients with a particular molecular profile to see if it works with them. Also so-called adaptive designs where you vary the enrolment of patients on the study according to the earlier results, and also including altering the randomization ratios and so forth. And also consider surrogates where you may not have evidence of net clinical benefit, but are there measures that you could use which would be an indication of activity? That's been fraught with problems in the past and requires some fairly sophisticated methods to say when that might be a way of undertaking it. So in cancer in particular, one can consider umbrella or platform trials where you look at looking at various mutation subtypes or molecular profile subtypes within one cancer. You can also consider basket trials where you might have one particular molecular profile across a lot of cancers and seeing whether that particular treatment might work for that basket of cancers. An example of that we've been undertaking here is the MOST program, which is Molecular Screening and Therapeutics, that David Thomas heads up, looking at modular single-seeking clinical trials. So what we've got here is patients who are screened, 1,000 initially, but we're looking at increasing that to, if there's other funding, up to 1,500. Having been screened with a panel and reviewed by a molecular tumour board, then enrolled into different modules, either according to a molecular profile for example, a CD4K6 inhibitor, a PARP inhibitor, or if there's no particular mutation, also considering enrolling patients on treatments such as immunotherapies. And then looking at measures at the end of that in terms of response, changes in tumour growth, and other surrogates such as circulating tumour DNA. If there are positive signals coming out of those particular profiles, such as if, for example, profile number 2 was positive, that could then lead to an expansion cohort. And if that was also positive, then looking at randomized studies against last standard line of therapy, which would require international collaboration with other partners undertaking similar designs. So in the future, I think we're going to see more of these clinical trials in personalised medicine where we're going to need great collaboration with basic science, industry, bioinformatics, biostatistics and trialists. It will require the development of reliable surrogates to pick up signals of activity to make it more efficient than simply waiting to see you've got numbers on overall survival, for example, which is a fairly crude measure of whether something's working and for which you really need randomised evidence. But we're still going to need randomised evidence, albeit hopefully on smaller sample sizes, to demonstrate that the treatments that do appear appropriate can go further. I'll skip that one. Just to make the comment about rare cancers, the FDA has now just made the first approval of a treatment which is determined according to a molecular profile and is agnostic to the particular cancer cell of origin. So this is going to be something seen more often in the future, but in the past you had to demonstrate the treatment was effective within your lung cancer or bowel cancer, whatever. I'm just going to finish by saying that we need to think of clinical trials in the context of a self-improving health system, where the clinical trials are a key component of work, but which will hopefully lead to improvements in healthcare and practice. But also, if we link that in with clinical quality registries, where we measure what we're doing, we can identify where there are gaps, where there are variations in care, and let's see that lead to a drive in the future studies. So, in terms of extending our evidence base for all patients in the future, it's going to be important to promote the value proposition of embedding clinical research in healthcare, to build and maintain capacity in clinical trials workforce and in clinical quality registries, to see that we have ways of coming up with more efficient designs for clinical trials, which both benefit investigator-initiated and industry-sponsored studies, and others will probably talk about how we can better embed clinical research in healthcare for seeing improvement in overall outcomes. And I'll finish there. Thank you.